Right, next speaker is Altieri. We'll be telling us about diversity, equality models. So, thank you.
Okay. So thank you very much. So I would like to thank the organizers for this invitation. It's a pleasure to be here. And today I would like to discuss one of my current projects, which is essentially based on the analysis of large complex systems through the prism of statistical physics. So this is a collection of works I have been collaborating with different people in Paris and abroad, especially at the University of Padua and the Max Planck Institute in Leipzig. And this is the outline of my talk. So first of all, I would like to give you the physical motivation, the intuition on why so many theoretical physicists are getting interested in high-dimensional theoretical ecology. And for doing that, I will stick to the generalized Lock-Volterra model with the random interaction. And starting with this generalized Lock-Volterra model, I will highlight connections with glassy-like phases and slow relaxation dynamics, which resemble somehow the aging dynamics that appear for people who are familiar in the audience with spin glass model to glassy system and structural glasses. And then in the second part of my talk, I would like to use this generalized Lock-Volterra model to provide a proof of concept for the applicability to human microbiomes, with a special emphasis on the gut microbiome. And I will use this generalized Lock-Volterra model to distinguish different physiological states in the gut that can be connected to different emerging patterns of the disorder generalized Lock-Volterra model. And then in the very last part of my talk, to make the connection with experiments, with empirical findings even more clear, I will look at production as a function of the biomass, and I will propose an alternative mechanism, a sublinear growth model, to be compared, to be in contrast and to be compared with the logistic model. And then I will get to my conclusions and perspective for future research. So you know, the field of theoretical ecology is gathering interest in recent years, reached by an explosion of experimental results and increasingly sophisticated techniques, but is also fostered and motivated by crucial questions that still need an answer. For instance, how does the diversity affect the evolution of the other species? What about cooperative patterns? Are these patterns ordered, disordered? How does an ecosystem respond to an external perturbation? And is it possible to quantify these response functions in a similar way as we do for physical systems? And also, is the same ecosystem more likely to relax to a single fixed point regime or to multiple fixed points? And what are the typical features of this fixed point? Are they stable, unstable, marginally stable? Well, part of this question have been successfully addressed up to now, but there is still much to be done, especially concerning chaotic dynamics in large ecosystems, how to quantify Lyapunov exponents, so the appearance of a chaotic dynamics, for instance, in a planktonic community, and also about rare event and cascade of extinction. So we know that in real ecosystem, there are heavy-tail distribution for describing, for instance, species abundances, but this is not, so we are not able right now to capture this heavy-tail distribution by using our models. So over the years, theoretical physicists, mathematicians have attempted to make sense to these ballooning variables by using different techniques. One of these is linear stability analysis, the other one is the theory of dynamical systems, but the true novelty now is that there is a growing focus, a growing interest on systems composed by an enormous number of variables that can interact in myriad ways. And so, keeping that in mind, we can use statistical mechanics tools and statistical mechanics of disorder systems to try to figure out these properties and to characterize emergent phases and collective behaviors in terms of phase transitions. So in theoretical ecology, there exist two benchmarks, two platform models. One is the MacArthur model, which is a research competition model that, as it is designed, as it is conceived, is just a competitive model. And on the other hand, we have a generalized Lock-Volterra model that I'm going to focus on, which has been introduced long ago by Locke and Volterra separately to describe this oscillation in the ocean, so for two competing species, prey and predator, but right now has become a cornerstone in a large class of complex systems, from complex financial market to evolutionary game theory up to ecology. And in particular today, I'm going to focus on this Lock-Volterra model in high dimension, This is the reason why I'm talking about generalized lockable Terra model. Well, the starting point is the dynamical equation for the relative species abundances, Ni, relative in the sense that these abundances have been normalized by the total number of individuals in the pool, and I is an index that runs from 1 to S, where S is the total number of species in the community. So the dynamical equation is basically a Langevin equation, which is described by a first self-regulation term that can be written as a derivative of a one-species potential that in the simplest possible scenario, in the logistic case, admits two fixed points. One in Ni equal to 0, that correspond to the extinction, which is an unstable fixed point, and another one in Ni equal to Ki that correspond to the carrying capacity, and is a stable fixed point. So why is self-regulation term? Well, because if a species I experiences overcrowding its immediate environment, it will be pushed back to its own value of the carrying capacity Ki. Then we have this other term that I'm going to detail in a while that accounts for interspecific effect. And that to complexify a little bit this setting, we introduce demographic fluctuations that scale as square root of Ni, so the square root of the population, satisfying detailed balance. And on top of that, we have also an immigration parameter that is crucial, is essential, because if you don't plug into the model this immigration parameter, because of demographic fluctuation, at finite time, every species will get extinct. So we need this immigration parameter to prevent an absorbing boundary in an I equal to 0. So the main assumption to get an exactly solvable model is to solve from a well-mixed community. So at this level, we don't have any space dependence, but we also generalize this approach in a meta-community scenario. So if you are interested, we can discuss more during the coffee break. And then, as I said, we introduce demographic fluctuation that can be modeled by a wide Gaussian noise with zero mean and variance given by this expression, where T is the amplitude of demographic fluctuation. So it basically plays the role of an effective temperature in a statistical mechanics approach. And the other point, which is the most tricky part of this approach, is what about this alpha ij? Well, as I mentioned, so this represents an open problem in theoretical ecology. So measuring the actual interaction in a diverse rich ecosystem is a very hard problem, is an open inference problem. But what we can do at the first level analysis is to follow Robert May, who was a pioneer in this field, who first studied stability and complexity in large ecosystems by using random matrix theory. And so we model these alpha ij as random coefficients that are extracted from a given distribution, a Gaussian distribution or any other distribution without long tails, that is parameterized by the first two moments, mu over s and sigma squared over s. So this scaling 1 over s is crucial in such a way to have a term here which is over the 1 in the thermodynamic limit. And then on top of that, we have this other parameter, gamma, that accounts for the degree of symmetry or asymmetry between these coefficients. So for gamma equal to 0, we get asymmetric interaction, but completely uncorrelated, which is a simple scenario. And for gamma equal to 1, we get the symmetric case, which is another simplified setting, because for gamma equal to 1, so for alpha j equal to alpha ji, this dynamical equation here, this Langevin equation, admits an invariant probability distribution that can be recast in terms of a Boltzmann-Gibbs distribution, exponential of minus beta h, and the properties of this h function, of this energy function, can be studied by using disorder system techniques. And so here we are. So this is the resulting phase diagram of the model, where the ultimate goal is to characterize emergent collective behaviors in terms of order or disordered phases, order or disordered phase transition. So here I'm plotting T, the amplitude of demographic fluctuation, as a function of sigma, the standard deviation of this random matrix. And so as you can see here, when the demographic noise is sufficiently high, the system is in the single equilibrium regime, meaning that the landscape of configuration is purely convex and is characterized by just one fixed point, a unique equilibrium. Then we can pretend to decrease T, the amplitude of demographic fluctuation, or to increase sigma, the heterogeneity of the interaction. And at a certain point, the system will cross this blue line, which is an analytical line, okay, so it's a rigorous analytical line, and will enter this multiply equilibria phase, which is characterized by this apparent two-level structure. So I also gave a characterization in terms of complexity or configurational entropy that accounts for the log of the number of equilibria at the given free energy density, average over the disorder, so over the alpha j, and evaluated in the thermodynamic limits. So for S, for the number of species going to infinity. And so between the blue and the orange line, these complexity curves are strictly positive. And so if you look at this expression, this implies that the number of equilibria is exponential into the system size, into the number of species, which is particularly timely if you think of stability landscape, ecological resilience concept that have been proposed over the years to explain this apparent emergence of alternative stable states in real ecosystems. And they have been also more recently resolved by Jeff Gore group at MIT in Boston, in which they consider artificial communities, so they plug into a chemostat under well-controlled chemical condition, different, so 48, 96 bacterial isolates from terrestrial environments or other kind of environments, and they could study it by changing the amount of nutrients or the dilution over different days, so 10 days. They could actually study biomass and the evolution of the abundances of the species. And so in these experiments, they could actually observe different regime. So a phase characterized by stable coexistence, where every species is there. A partial coexistence where some species get extinct, and another phase characterized by persistent fluctuation that include limiting cycles or also chaotic dynamics. But from a theoretical standpoint, the other interesting part is that if you keep decreasing t, at a certain point, the system will cross this other instability line, this orange line, and will enter a Gardner phase. I call it Gardner in analogy with glassy system and amorphous material. And so this means that each of these basins of attraction splits, shatters into many sub-basins according to a fractal and hierarchical structure. And so now the next question is, okay, but what about plugging a small degree of asymmetry into the model? So in switching on a small degree of asymmetry will correspond to introduce into the model dissipative forces. So it will correspond to the dissipative forces in your dynamics. And so if you switch on a small degree of asymmetry, these two phases will persist, will remain there, because the equilibria are globally stable and locally stable. So this one will be replaced by an exponential number of chaotic attractors. But the last one, the Gardner phase, will be completely washed out, wiped out, because the equilibria are marginally stable. And so it's sufficiently to add a small asymmetric perturbation to completely wash it out and destroy this phase. And now that we have a complete picture in terms of the thermodynamics, we can wonder, okay, but what about dynamics? What we can state in terms of the dynamics? Well, we use here a dynamical mean field theory approach that allows us to move from a many-body involved problem, so from the Langevin equation that I showed before, to a self-consistent stochastic process for the single degree of freedom, that is the mean abundance, N of t, so a coarse-grained variable that describes the mean abundances of the species. And so there is a price to pay to move to this dynamical mean field theory approach, because in this self-consistent stochastic process, now this variable is coupled to an external path. And the statistical properties of this external path have to be determined in a self-consistent way. But we could study the dynamics, in particular in the single equilibrium regime and in the multiple equilibrium regime, and this is what happened. So in the single equilibrium phase, if you look at dynamical correlators, so N of t prime, average over number of samples, initial condition, demographic fluctuation, and alpha ej, so the disorder interaction, and you plot these dynamical correlators for different t prime value, you can see that all curves collapses to the same value, which is this Q0, which is the theoretical predicted value in a disorder system approach. So for people who are familiar with disorder system, in a replica computation, in a replica approach. And so this curve here satisfies a time translational invariant property in the sense that dynamical correlator does not separately depend on t and t prime, but just on a single time scale, t minus t prime. And so the dynamics here is time translational invariant. On the other hand, if you look at the same curve in the multiple equilibria phase, the situation is dramatically different. So the dynamics never sit down in any of these equilibria, but start to explore, to wander across the most numerous and marginally stable direction, exactly as it happened in glassy system. So this reminds us in aging dynamics, that means that the longer you wait, the longer the system takes to decorrelate. And so by looking at this rescale correlation function, you can see that never collapses, never decay to zero, but start displaying two different plateaus that correspond to Q1 and Q0, so the two order parameter that I showed before in the phase diagram. So the size of the largest basin of attraction and the size of the deepest basin of attraction in this apparent multi-level structure. So this is for the dynamics. And so from, as I said, from a purely theoretical standpoint, for a purely theoretical perspective, but now I would like to make a connection with experiments. And in particular, I would like to stick to this single equilibrium phase and to use this disordered generalized local algebra model to try to interpret metagenomic data for the gut, for the human microbiome. So this is a work in collaboration with people at university from the interdisciplinary lab at University of Padua, in particular Samis Weiss and gastroenterologists from University of Padua. So here we are looking at order abundance as a function of samples for two different cohorts, healthy and unhealthy patients, where for unhealthy I mean patients that are affected by different syndromes, so inflammatory bowel disease that include ulcerative colitis or Crohn's disease, which is a chronic inflammation of the gut that can have severe implication also on autoimmune system, malnutrition, weight loss, and so on. And so you know the taxa composition is usually obtained by inference techniques, so advanced inference technique that allow for the characterization of ribosomal RNA genes or for the reconstruction of the entire shotgun sequencing. Here we're using a novel algorithm that was conceived by Samis Weiss and Lagomarsino in 2021 that is called a core-calgio algorithm that allows us to have access not only to taxonomic grouping but also to species abundances. So species abundances of a large set of species. And so as I said here, we are looking at OTU, so order abundance as a function of samples, where for samples I mean different individuals, different patients, and then comparing healthy with unhealthy. And so this is a Mueller plot for the single temporal snapshot. So from this Mueller plot, we can extract the mean abundances of the species, but also high order correlations that appear to be particularly convenient, particularly appropriate to be compared with theoretically motivated order parameters in a disorder system approach. So the next step, we just require to define our sampling protocol. So to figure out how our sampling protocol works. So we have access to two different quantities. One is S, the number of species, okay, in each cohort, and the other one is R, independent realization of, so R, independent and correlated observation of our reference ecosystem. So this is from empirical data. So we interpret the average over species as an ensemble average, and the average over R independent realization as a disorder average, and which is now denoted with the upper bar. So we measure this quantity from the data, and in particular here we have H, which is the mean abundance, Q0, which is the interstate overlap, as I said before, and QT, which is a self-overlap. So these are high-order correlation. And as you can see from this empirical data, so from this parametric plot, there is a sharp separation, so data for healthy and diseased patients, respectively in blue and red, are well separated, are clustered. So forget about the gray dot that correspond to a null model, while sample labor have been just randomized. So I can go into detail deeper later, but I would like to focus for the time being just on the blue and the red dot, that correspond to healthy and disease. And so you can see that are really clusters. So there is a true separation between healthy and unhealthy patients. And so the next step, as I was saying, is to infer the interaction in this model. So we need to define a non-convex optimization theory for this model, where the optimal parameters, so the extrema of a given cost function, correspond to the optimal parameter in an inference, a non-convex inference problem. And so here we define this cost function in this way. So this cost function provided the information on pi, so on the mean abundance, high order correlation and carrying capacity, so the maximal level of the species, so the average of the maximum species abundances for each cohort, we extract information on this vector, theta, that encodes the interaction parameters, so mu and sigma, the noise amplitude, and the migration parameter. And so we are not guaranteed that this cost function has a unique solution, and this is the reason why we define it as a combination of square relative errors, where delta H, delta QD, and delta Q0 account for the square relative errors, so the difference between the estimated order parameter from the model and the one obtained measured from data. And then we perform a greedy search in such a way to recover this theta star, so the optimal vector of parameters that correspond to the interaction matrix parameters, the noise amplitude, and the migration. And so this is to recap our protocol. So we started from a generalized local thermodynamics, and provided that these dynamics converge to a stationary states, we can perform two different kinds of averages, one over species, that is an ensemble average, and another one over samples, so over independent realization, that is a disorder average. We measure this quantity, H, QD, and Q0 from data, and then we infer these parameters. And with that in mind, so with the early assumption that community in the same microstates share the same statistical properties, we can reconstruct the phase diagram for the inferred parameters. And so here I'm plotting the demographic noise amplitude as a function of the heterogeneity of the interaction. And again, you can see that data are really clustered between healthy in blue and unhealthy in red. So darker dots do not correspond to patients because these are already average over number of species and number of samples, but correspond to the solution that are associated with the lower value of the cost function. Whereas the hexagonal marker is the ground state. So it's the lowest value ever of this cost function. And again, you can see that there is a clear separation between healthy and unhealthy. And in this set, I'm showing the species abundance distribution at fixed disorders, so for fixed patient Z. But since we have information also on the alpha ij, so our ultimate goal was to infer the interaction matrix parameter, mu and sigma, we could also plot the frequency P of alpha as a function of the interaction value. And so even though they are both described by a Gaussian distribution, you can see that in unhealthy patients, the sigma, so the heterogeneity of the interaction, is much smaller, as a signature of the fact that these biases tend to reduce the heterogeneity of the interaction. So now you can wonder, what about stability? So does it mean that healthy cohorts are more stable than the other? Yeah, the answer is yes. So we also compute the leading eigenvalue of a stability matrix, so a Hessian matrix of the free energy. So we diagonalize this matrix and we could have access to this leading eigenvalue. And in particular, we could see that healthy cohorts are unaffected by external perturbations. So I have a leading eigenvalue which is pretty close to 1, has a clear signature, a clear evidence of stability, whereas unhealthy cohorts are characterized by an eigenvalue which is scattered all over the parametric plot and is going deeper and deeper, so it's getting closer to 0, which is the stability, the marginal stability threshold. So this means basically that unhealthy cohorts are more affected by external perturbation or antibiotic treatments and so on. So this is just a proof of concept. So as I was saying, we are sticking to the single equilibrium regime. So it would be nice for the future also to analyze what happens in the multiple equilibria phase, and if these unhealthy patients are really characterized by a multiple equilibrium regime, but this finding also paved the way for developing new controlled and therapeutic strategy in tuning microbiome host interaction. And then in the very last 5 minutes, I would like to describe another model. So as I said at the very beginning, so a long-standing question concerned this diversity stability debate. So what is diversity and what is stability is a very hard question to answer, but the problem so let's say, ecological theory has long predicted that more diverse ecosystems are intrinsically unstable. And so if you look at my phase diagram before, you can see that when you increase sigma, when you increase the heterogeneity of the interaction, the system becomes more and more unstable. So ecological theory that is based on generalized Locke-Volterra model with logistic growth and the result of Robert May, as I was saying, that is a pioneer in this field, predict that diverse and more diverse ecosystems are intrinsically unstable, which is somehow puzzling, astonishing if you think of the great diversity that is observed in nature. And so we try to propose an alternative mechanism to be compared with the logistic growth. And in particular, everything originated from this paper by Yanaton, McCann, dating back to 2015, in which they studied, they observed the biomass across terrestrial and aquatic biomes, terrestrial and aquatic environments, and they could see that in prey-predator biomass, there is a power law behavior. And so if you look also at production of the overall community as a function of biomass, you can recover a power law behavior, so with an exponent that is smaller than 1. So this was somehow puzzling, and no model today could explain this kind of behavior, and in particular this emergent stability when you increase the diversity, when you increase species richness in your ecosystem. So this was our goal. So we collected data across different community types, from mammals, phytoplankton, trees, up to marine bacteria, for n separate ecosystems. So small n is the number of separate ecosystems for each community. And so we look in log-log scales at production as a function of biomass. And so you can see here that growth is found to scale sublinearly with biomass. So what is missing? So the missing piece is a theoretical rigorous model that could explain this, and in particular that could provide an alternative mechanism to this diversity and stability positive relationship. And so we propose a theoretical framework to show that competitive interaction can be collectively stabilizing with diversity. So this is what we term a sublinear growth model, to be compared with the logistic model. So you can see here that even though at the single species level, at individual level, logistic model seems to be stable and to saturate here, when you increase the diversity of your ecosystem, so when you start increasing S, the number of species, logistic model appear to be unstable, whereas the sublinear model appear much more stable. And so we term it sublinear model because it's described, is defined by sublinear growth term plus a linear loss plus the same mass action law as the logistic model in such a way to be on the same footing, on the same scale. And so how is it possible? Yeah, we also study the probability of observing a stable system as a function of species diversity. And so you can see here that for the logistic model, the probability of observing a stable system as species diversity increases decline to 0, whereas in the sublinear model increases to the upper, so to the maximal value, which is 1. Then we look at the community matrix or Jacobian matrix. So we plot the imaginary part as a function of real part of the eigenvalue in the complex plane. And so a criterion for stability is that all the eigenvalue of your community matrix have to be negative, have to have a negative real part. And so what happens here in the logistic model, the smallest eigenvalue is pushed towards 0 as a signature of instability when you increase the number of species, whereas in sublinear model, you observe the opposite scenario. So the smallest eigenvalue is pushed away from 0. And then we generalize this result for random interaction matrix. In particular, for a matrix AAJ, which has a strictly positive mean, in such a way to enforce the interaction to be competitive, and a generic standard deviation. And you can see here, again, that in the logistic model that is plotted in blue, stability resides below this uniform line, whereas in the sublinear model, stability resides below this full orange line, which is a never increasing stability boundary at variance with the logistic model. So the full orange line corresponds to theoretical prediction that have been obtained by disorder system and cavity computation, whereas the color shading corresponds to numerical simulation. So this is to give you a complete picture and also to contrast the sublinear model, which has been introduced also for explaining individual ontogenetic mechanisms to the logistic route. And with that, I would like to get to my conclusion for the last 5 minutes. So I present a generalized lock-Volterra model in a well-mixed approximation, so no space dependence, that incorporates demographic noise and heterogeneous interaction, so random symmetric interaction. Even with symmetric interaction, we could detect 3 different phases: a single equilibrium phase, a multiple equilibrium regime, which is characterized by this apparent 2-level structure, and an amorphous garden-like phase, which is marginally stable. And so if you look at the dynamics, you can also associate with these 2 last phases to a slow relaxation dynamics, which is called aging, as I said. So the longer you wave, the longer the system takes to decorrelate, which resembles somehow spin-glass problem and glassy systems. Then in the second part of my talk, I presented a data-driven approach for inferring the interaction from the human gut, and in particular to distinguish different physiological states and associate them with different interaction pattern of the disorder generalized lock-Volterra model. And then in the very last part, I presented this sublinear growth model to try to interpret production as a function of biomass. And then some perspective for future research. So I would like to deep investigate this multiple equilibrium phase for microbial communities by combining numerics with tailored experiments. Then another interesting perspective will concern this untangled research species dynamics and how to go behind the logistic growth model, and then perform also a renormalization group approach for spatial models in finite dimension. So with that, I would like to thank all my collaborators so far, and I would like to thank you for your attention.
Thank you. Fascinating talk. Question for Ada Altieri. Okay. So, yeah.
Can I refer to the last part of your talk about diversity versus biomass? I mean, scanning quickly the exponent that you put there, and you didn't give any error bars on these, but they all hover around 3/4.
Yes, exactly.
It may suggest some effective medium or dimensional analysis, simple dimensional analysis that can lead to that.
Yeah, exactly. So all the exponents are between 2/3 and 3/4, and this is the reason why when we did the analysis, the theoretical analysis here, for instance, so this slope here can depend on the exponent k that you put. So for our theoretical analysis, we assume k equals 3/4, but then you can try also to give an explanation, let's say a practical explanation of this exponent, because actually the sublinear growth represent an unresolved question in theoretical ecology. So it's not known. So the mechanism behind this sublinear growth is unknown. So some people suggest a sort of animal grouping behavior that has a reproductive skew. And so if any group has a reproductive skew, when you look at the overall community, you should observe sublinear growth. But another explanation is related to geometric constraint, surface versus volume constraint, that can explain this sublinear growth, and is also to be the main mechanism of self-tunneling rules in forest. So, yeah.
Great question. So in the first part, we had a random Lotka model that so I was thinking in the ecological models, you often have a hierarchical structure, right? That one layer mostly couples to the next layer, and so you, because I mean, one species eats like another species and so on. Does that more like correspond to different types of matrix,
I haven't looked at that, so I'm just considering a well mix. So every species is connected with all the others, so it's a fully connected model. But there are people in other communities, so for instance, Tobias Gala, who have looked at this kind of structure. So you can look, for instance, at the hierarchical structure of the interaction and to see what happens. So it's interesting, especially because with this kind of interaction, you can model trophic levels, and you can also recover the empirical species abundancy distribution.
Exactly. I mean, this is definitely nice as a theory.
Yeah. There are people working, on, No, makes sense. Definitely makes sense. And so depending on the structure or your alpha J, you can recover a species abundancy distribution, which is more consistent with data. So, yeah, because here I can, let's say with this normal distribution, I can basically do all the computation. So as a first level analysis, I assume that, but I perfectly agree that you can have different kind of interaction. So this one again, it's a well mixed model. So with no space dependence, no structure, but then in a generalization, here, so sorry, I have to go to my references. So here in this paper, so in this PRX Life, we consider a meta-community scenario. So that means that we are coupling different patches. And so we consider a correlation between different patches. And so this is somehow a sort of, so it's a way to introduce space dependency in your model and to study correlation between different patches. And so there, depending on demographic fluctuation and dispersion rate, you can detect different phases. So a continuous phase transition or a discontinuous phase transition. So.
Thank you. Okay, I mean, so in the gut biome, do you know if there's different trophic levels or how heterogeneous, basically heterogeneous should your gut biome be? Or do people know? Okay.
With hierarchical interaction, you can actually model different trophic levels or.
But are different trophic levels present in the gut?
I think so. I don't. Yeah.